Now, the second part, I'd like to focus on the actual process of DNA replication. And the objectives are as follows. At the end of this section, the student should be able to articulate the very simplified version of what we call the central dogma of biology. The student should be able to identify the model that proves the process of DNA replication, and kind of discuss a little bit about the experiment. And the third objective is that the student should be able to articulate all the 7 or 8 enzymes and elaborate on the function of each one of these enzymes. Now, we won't dig too deep into mechanisms until we get to the next lecture and beyond. Let's talk about central dogma. Central dogma of biology is this process where it goes from DNA is replicated to form other DNA molecules. The DNA is transcribed to make RNA. RNA is translated to make protein. And as you all know, the protein are the workhorses of the cells right here. And that basically is the central dogma. We're starting with doing the DNA replication right here, and then this will follow at the end of this semester with the transcription, and then next semester we're going to devote the entire semester to the translation process and protein synthesis. So, DNA replication. How is it that you go from one DNA molecule to 2 identical DNA molecules? No change, no alteration. There were 3 models that were proposed. There was a conservative model, semi-conservative model, and dispersive model. I'm going to ignore the dispersive model because this was immediately unaccepted, but we're going to focus on the conservative. And if it were conservative, you will have 2 bands. One band will be N15, N15. The other band will be N14, N14. That's if it's conservative. If it were semi-conservative, take a look at it. This is the parental DNA leading to the formation of 2 daughter DNAs, right? Each one consists of N15 and N14. The other one is N15, N14. These have the same density. They are neither light nor heavy, but they are in the middle. We call it heavy light. One band. 2 generations. Yes. 2 bands. Heavy, light, right here, and light. That's exactly what we see. Okay. And go ahead and look at the others. Let me say this before we begin into the actual DNA replication itself. The DNA replication must take place for cell division to occur. We know that it's very closely linked to cell division. These are the phases of cell division. Mitosis is where the cell divides, but you have G1 and S is where the DNA is synthesized. You have G2, and before they go, the cell goes into the mitotic phase. Okay? All right. So, DNA replication. What makes it possible for a DNA to go from one molecule to 2 identical molecules that are identical to the parental DNA? Single-strand binding protein binds. Then we're going to cover the leading strand and the lagging strand. Leading strand is synthesized in one continuous fashion, and that is an enzyme called primase synthesizes a short RNA primer. To lay it down, DNA polymerase 3, then using the 3' OH group, it begins to build nucleotides onto nucleotides by adding it and extending that in one continuous fashion until the DNA strand is completely copied. Now that is the leading strand. The lagging strand, on the other hand, because of stoichiometric problems. It cannot do that. Okay? Now, please remember that, and we're going to talk about this a little bit more, in that DNA polymerase that actually generates synthesizes the DNA strands, they only work in the 5' 3' direction. And so it's okay for the leading strand to be synthesized that way. As the replication fork opens up, that strand continues to be made through the replication fork. Stoichiometrically, it's not possible for the other strand, and we call this the lagging strand, because it's sort of going backward. It still goes from 5', 3', but it's moving backward. And I'll explain it a little bit more. And I know you all remember this. So what happens with the lagging strand? Well, the primase again synthesizes the RNA primer. DNA polymerase 3 then comes in and extends that. Now, since it's not possible to do it in one complete fashion, it does it in sections, which means that instead of one RNA primer being laid down, like for the leading strand, it lays down quite a few RNA primers as the replication fork opens up. So RNA primer is laid down by the primase, DNA polymerase 3 extends it. DNA polymerase 1 comes in and chews up the RNA primer because it doesn't need to be there in the final DNA product. And then as it chews it up, it fills in the gap, but it cannot completely connect the DNA fragments, and that's the job of the DNA ligase. DNA ligase comes in and connects the DNA fragments. And now you have two complete DNA molecules that are made from one DNA molecule. Okay? There are two problems that we need to address. Number one, as we stated, the strands are copied in both directions. The synthesis goes from 5', 3', and they're copied 3', 5'. And as I pointed out, there are no known DNA polymerases that work in the opposite direction. One strand can go from 5', 3', but the other strand, how can it go in the opposite direction? Well, it can't, and it doesn't. And it's worked out by Dr. Reiji Okazaki, who used Paul's experiment to label the DNA, grow E. coli on tritiated thymidine. And tritiated thymidine will change the weight of the DNA, okay? The DNA from 7S, and these are sedimentation values, to 11S. Okay, he called this the Okazaki fragments. These are short stretches. Okay, and then he did pulse chase, and that is that he transferred the E. coli to unlabeled media, and then he looked at the sedimentation, the pelleting of these Okazaki fragments, and what he found was this: that Okazaki fragments change their sedimentation coefficient. In other words, they get incorporated into larger and larger DNA fragments as the chase continues to be lengthened. In other words, you have short fragments that are synthesized, as we stated, right? And then DNA polymerase 1 comes in and chews up the RNA primer, and it begins to fill in the gaps, and you begin to extend. So the longer that you incubate that, the longer that the DNA fragments from the lagging strand will look. The Okazaki fragments begin to be joined to each other. And so this kind of proves that one strand is synthesized in a continuous fashion, while the other strand is synthesized in discontinuous fashion. This is just a picture of the Okazaki fragments, and this is, take a look at it. This is the replication fork. This is the leading strand. So you have RNA primer that will be laid down, and then DNA polymerase III comes in and synthesizes this. As you open this up, it zips all the way through without any problem. Now, in order for this to be synthesized, what I said was, if there was a polymerase that will synthesize this from 3' to 5', that will be perfect. That won't be a problem, right? But there are no DNA polymerases that will do that. In order to make sure that that happens, you're going to do it in sections, and as this opens up, you lay down more primers, DNA polymerase III comes in. As you open this up, more primers, DNA polymerase III, and then after DNA polymerase III synthesizes DNA using the RNA primers, then DNA polymerase I comes in, chews up the RNA primer, fills in the gaps, and DNA ligase comes in and closes it up. Problem number 2, and we just addressed that, and that is DNA polymerases cannot initiate DNA synthesis de novo. Okay? Again, they found that the Okazaki fragments contain RNA regions that were complementary to the DNA template. And what are these RNA regions? Well, the RNA regions consist of the RNA primers. And so, in order for DNA synthesis to occur by DNA polymerase III, it must have a 3' OH to use in order to build onto. And that 3' OH is provided by the RNA primer that is laid down by the action of the primase enzymes. There are actually 2 RNA polymerases that generate primers. One of them is a large one, RNA polymerase, and we can call it DNA-dependent RNA polymerase. It is large, 450 kilodalton. It is responsible for generating RNA primer in the leading strand. This is sensitive to the antibiotic rifampicin. The other one is a smaller one, primase, which is 60 kilodalton, and it is responsible for generating RNA primer in the leading and also the lagging strand. And this one is resistant or insensitive to the antibiotic rifampicin. Now, if you were to treat a strain of bacteria with rifampicin, what you will inhibit is really, you will inhibit the synthesis of the leading strand, but the lagging strand will not be affected because the primase is not affected by rifampicin, only the polymerase. Important for the following reason. You remember that DNA polymerase I has to come in and chew up the RNA. It chews up the RNA from the 5', 3' direction, and this is what this is, right? It chews up the RNA from the 5', 3' direction, fills in the gaps, and DNA ligase comes in and joins the DNA fragments. Now let's talk about very quickly about the DNA replication itself. DNA replication begins with an origin of replication that is very AT-rich right here, where you have opening of the replication bubble, and in the eukaryotic DNA, it occurs at 150,000 at every 150,000 base pair region. The replication origin is recognized by a protein called DNA-A protein. DNA-A, multiple proteins of this DNA-A will recognize that replication origin again, which is very AT-rich. And they will recruit other proteins, and the other proteins that are recruited is DNA-B, which stands for the helicase enzyme. Okay? But other proteins are recruited here. And so what happens is the helicase comes in and it begins to open up the replication fork, and DNA replication begins. And we're going to go through each one of these. This is in the prokaryotic system. It's a little bit more complex for the eukaryotic system, but they're very, very similar. So the first thing is that helicase comes in and breaks up the hydrogen bond and open up the replication fork. DNA gyrase moves in front of it, right, and begin to relax that to make sure that that replication fork proceeds through, so DNA replication can continue. This is the helicase enzyme. Of course, in order for it to break up the hydrogen bond, this is an energy-dependent process. Then as the DNA strands are separated, they are stabilized by single-stranded DNA binding proteins. Now, this serves two functions. Number one, to keep the DNA strands single-stranded, separated, right? It is possible that they might come back together. That reaction is very favorable, or it can form double-stranded loops within itself. And so the single-stranded DNA binding protein serves the purpose of keeping it intact, single-stranded. The second purpose it serves is that it also keeps those two single-stranded DNA from being cleaved or being digested by nuclease enzymes. This is a picture of single-stranded DNA binding protein. Without the DNA binding protein, DNA polymerase will read through, and then it will stop right here, where you have loop formation of double-stranded regions right here. So that's why the single-stranded binding protein binds to it to keep it single-stranded. Then the primase comes in and lays down a primer. It lays down primers, multiple primers for the lagging strand and one primer for the leading strand. DNA polymerase III comes in and using the OH that is free on that RNA primer and begins to synthesize stretches of DNA in the 5' 3' direction. As we say, DNA polymerase III synthesizes one time all the way through for the leading strand, different stretches for the lagging strand. The DNA polymerase is a processive enzyme. In other words, it stays on the DNA and it doesn't fall off until the time has come for it to fall off. And so we call that processive. Now, what enables that is there are two proteins. There's a clamp loader and a DNA polymerase clamp right here. And really, DNA polymerase clamp is exactly what it says. It actually goes around as a clamp around the DNA and anchors the DNA polymerase to that region so that DNA polymerase can go along the DNA molecule as it synthesizes the new strand and without falling off. Again, this is just a picture of DNA clamp, and when the time comes, when it reaches a point where it needs to fall off, then it falls off, and it is used for another synthesis. DNA polymerase 1 comes in, and it begins to chew off the RNA primers right here, and then it fills in the gap, right, using the free OH on the other side right here, and it fills in the gaps, and then this is followed by DNA ligase that comes in and join all the DNA molecules. Again, this is an ATP requiring reaction, and this is the mechanism of the DNA ligase. Now, let me just say this. This is the final slide, and it kind of shows you right here all the enzymes that are involved. Now, I know this is kind of late, and I'm sorry that this is late, and I apologize for uploading, but we're still trying to catch up since we're down one faculty member. But tomorrow we're going to go through some IRAD that are going to be very straightforward, and then we're going to go through some TRAD for teams, and the TRADs are not going to be as laborious as they will be later on, but they're going to be a few questions that will require the groups to work together. Remember, tomorrow is when we begin to grade this. 25% will come from the IRAD and 25% from the TRAD. Thank you, and we'll see you all bright and early tomorrow morning.